Okay, now we embark on our adventure through the small intestine. In this first section, we'll consider the anatomical features as well as intestinal motility. And then in the next section, we'll move on to the chemical digestion and absorption that occurs in the small intestine. So let's begin here, where nearly all the chemical digestion and nutrient absorption occurs. It's the longest part of the digestive tract for sure. It can be up to 4.5 meters long in a living person, but when you lose all the muscular contraction in a cadaver, you'll see that it could be up to about 8 meters long. And certainly the word small refers to its diameter, not its length. The small intestine is really highly convoluted and coiled. It fills most of the abdominal cavity, and it exists inferior to the stomach and liver. Part of it is retroperitoneal, and part of it is intraperitoneal. It's almost as if the intestines were pushing through the peritoneum to become intraperitoneal, but only some of it made it all the way through. So some is wrapped in serosa, and some is retroperitoneal and thus not. So the small intestine is divided into 3 regions: the duodenum, the jejunum, and the ileum. The duodenum, or duodenum, duoden meaning 12, which referred at the time to about 12 fingers of length, which is about 10 inches, begins at the pyloric valve. The pyloric valve is the valve where we move from the stomach into the small intestine. And there are major and minor duodenal papilla there, where we'll see the entrance of the bile duct and the pancreatic duct at the major duodenal papilla, and the accessory pancreatic duct at the minor duodenal papilla. The small intestine arches around the head of the pancreas at the duodenal jejunal fissure. Most of the duodenum is retroperitoneal. It receives the stomach contents, chyme, pancreatic juice, and bile. This is where stomach acid is neutralized by many of the pancreatic juices. Fats are physically broken up and emulsified here by the bile acids, and pepsin is activated as the pH becomes more alkaline. Pancreatic enzymes take over the job of chemical digestion at this point. The jejunum is the first 40% of the small intestine beyond the duodenum, and it gets its name because the root jejun implies empty or dry. Usually upon investigation, this area was empty, so they named it the jejunum. It's about 1 to 1.7 meters in a living person. It has large, tall, and closely spaced circular folds, and the wall is relatively thick and muscular. It's especially rich in blood supply, which allows for most of the digestion and absorption to occur in the jejunum. The final section is the ileum. It forms the last 60% of the post-duodenal small intestine. It's about 1.5 to 2.5 meters or so. It's much thinner and much less muscular. It's less vascular for certain. And it's very twisted, hence the name ileum. The root ileos means twisted. This is where we find the Peyer's patches. Remember those lymphatic nodules that form clusters on areas of the digestive tract? Here's the site we find them. If we were dissecting cadavers, you'd find that they were readily visible with the naked eye, and that they become progressively larger as we approach the large intestine. So this is where the lymphatic system has a chance to check out the safety of the materials that are traveling through the intestines. Where the ileum joins the large intestine is called the ileocecal junction. The cecum is this pocket of the large intestine sitting right here. The ileocecal valve is the sphincter here that regulates the passage of food and other digestive residue into the large intestine. It's formed by a thickening of the muscularis on the ileum. Both the jejunum and the ileum are intraperitoneal, and thus they are covered with serosa. So now let's take a closer look at the anatomy of the small intestine. The tissue layers are very reminiscent of those that we see in the esophagus and stomach. However, they have modifications for nutrient digestion and absorption. The whole lumen is lined with simple columnar epithelium and goblet cells for secretion of mucus substances. The muscularis externa has a very thick inner circular layer and then a much thinner outer longitudinal layer. The small intestine has a vast surface area. If you were to look at it like the inside of a garden hose, then the surface area would just be about 0.3 to 0.5 meters squared, which isn't that much. But by the time we see these three mechanisms in place, there's about 200 square meters of surface area. We acquire this additional surface area through three mechanisms. First, the circular folds are going to increase the area by a factor of 2 to 3. And then we'll see villi, little projections, finger-like projections all over the inner surface, which will increase the area by a factor of 10. And then microvilli all over the surface of the villi increase the surface area by another factor of 20. Let's look at those three mechanisms. First, let's look at the circular folds. The circular folds are like 10 millimeter high wrinkles all the way down through the small intestine. They're very prevalent in the duodenum. However, by the time you get to the ileum, they're quite small and sparse. And they act to create spiral action in the chyme as it flows down through all the convolutions of the small intestine. And that allows much more contact of the chyme with the mucosa and promotes thorough mixing and adds to nutrient absorption. Most of the absorption is done before we get to the ileum, so that probably explains the fact that there are less circular folds down in the ileum. Now look at the villi. The villi, in addition to the folds, we have villi between and on the folds. They're maybe 0.5 millimeter to 1 millimeter tall. They make the mucosa look kind of fuzzy. The villi have the epithelial cells all over them. The epithelial cells are a mixture of the absorptive cells or enterocytes, normal columnar epithelium you see in pink, and then goblet cells. And goblet cells will secrete the mucus. These are the cells here in blue. All these epithelial cells are joined tightly by tight junctions, just as we saw in the stomach, which prevents the digestive enzymes and acidity from seeping between the cells and digesting ourselves. The core of the villus is filled with the lamina propria, or tissue of the lamina propria, which is predominantly areolar tissue. Within this areolar tissue, we'll see an arteriole, we'll see that spread into a capillary network and exit through a venule. We'll also see a lacteal up into each microvillus. The blood capillaries of the villus are going to absorb most of the nutrients, and the lacteal will take most of the lipids. As we observed before, it will create sort of a milky looking substance in the lacteals because it's predominantly fat. The microvilli are on the apical surface of each absorptive cell. So you can see them very finely pictured here, microvilli on the top of each epithelial cell. And those are only about 1 micrometer high. And we call these microvilli the brush border and really increases the absorptive surface area. The brush border contains enzymes within the membranes of the microvilli. So chyme needs to move close to those enzymes because the enzymes don't come away from the cell membranes. And they carry out digestion right there in the brush border. This is called contact digestion. So it's very important that there's intestinal churning of the chyme. In addition, there are a few smooth muscle fibers we'll see within each microvilli that contract and help the lacteals empty, as well as helping circulate chyme. At the very bottom of the microvilli, we'll see these intestinal crypts. They're numerous pores that open up into tubular glands. And being glands, of course, they're going to have some secretions. They're very similar to gastric glands. In the upper half, the enterocytes and goblet cells seem very much like villi. But in the lower half, it's dominated by dividing stem cells. The lifespan of a cell is about 3 to 6 days. So new epithelial cells will migrate up out of the crypt, and they'll be pushed all the way to the top of the villi within 3 or 6 days. There, they're sloughed off, and then they'll be digested, just like any other material that passes through the small intestine. We'll also see a few Paneth cells clustered at the base of each crypt. The Paneth cells are going to secrete things like lysozyme, phospholipase. They'll also secrete defensins, and defensins are defensive proteins that will resist bacterial invasion of the mucosa. In addition to the Paneth cells and defensins, we'll also see that there are defensive lymphocytes throughout this lamina propria within each microvillus. And these defensive lymphocytes are going to intercept pathogens before they can invade the bloodstream. They'll sometimes aggregate into lymphatic nodules, which we see in the ileum as Peyer's patches. Here we can see some scanning electron microscope villi, as well as a histological section, which shows all the intestinal crypts, as well as the villi, duodenal glands, and then this thick layer of muscularis interna. So intestinal crypts will secrete about 1 or 2 liters of intestinal juice per day. They secrete this juice in response to acidic conditions, hypertonic chyme, and distension of the intestines. The juice is a pH of about 7.4 to 7.8, and it contains water, mucus, and a little bit of enzymes. But most of the enzymes that function in the small intestine are found at the brush border and in the pancreatic juice. So there are a couple of different means of intestinal motility. These contractions of the small intestine serve a few functions. First of all, they're going to mix chyme with intestinal juice, bile, and pancreatic juice, which acts to neutralize the acid and digest nutrients much more effectively. The contractions also churn chyme and bring it in contact with the mucosa for contact digestion and nutrient absorption. And finally, the contractions aid in moving residue towards the large intestine. Segmentation is the first means of intestinal motility we'll look at. Segmentation is the alternate contraction of ring-like constrictors along the length of the intestine. As one relaxes, a new constriction forms elsewhere. You can see in this figure these alternate waves of contraction, and the purpose of them is to really mix and churn material, not so much to move them along. We'll see that when we look at peristalsis. The segmentation is governed by these pacemaker cells that are in the muscularis externa. They set the rhythm. In the duodenum, it's about 12 times per minute for each stationary constriction to contract and relax. It goes down to about 8 or 9 times per minute in the ileum. Because of this difference in the rate, there's a slight bit of pushing of materials through the small intestine. When most nutrients have been absorbed and little remains but the undigested residue, segmentation will cease and peristalsis will begin. Peristalsis is a wave-like contraction that we saw in the esophagus to push a bolus of food down into the stomach. This peristaltic wave is going to begin in the duodenum, and it'll travel about 10 to 70 centimeters and then die out. And another wave will begin slightly downstream of the first. That acts to push material through the small intestine. It's known as a migrating motor complex, which is successive overlapping waves of contraction. They basically milk the contents of the small intestine towards the large intestine. Once they reach the end of the small intestine, we come to the ileocecal valve, which is usually closed. It's the gateway between the small intestine and the large intestine. However, food in the stomach will trigger the gastroileal reflex, which enhances segmentation of the ileum, so churning of food, and then it will relax the ileocecal valve. As the cecum fills with residue, pressure pinches the valve shut and stops further movement of material into the cecum and prevents backflow from the cecum into the small intestine. Okay, now we've finished the small intestine. We're ready to go on to chemical digestion and absorption.